I am very excited to present, introduce our speaker today. Alejandra Verdugo. She did her bachelor's at UNAM in Mexico, and she did her PhD with Brandon Gout at UC Irvine. Then she did her postdoc at ETH Zurich with Martin Ackerman. Now she is faculty at UC Irvine, so not very far from us, and she's been doing some really amazing and beautiful and exciting work on microbial communities, and we're very thrilled to get the chance to hear her. So please help me welcome Alejandra. Thank you.
Thank you. Thank you, Pam, for this very kind introduction, and I'm very happy to be here. Thank you, Pam, and all the department for hosting me today. I will be talking about the work we have been doing in the last 5 years in my lab at UC Irvine. And this is really work trying to use simplified microbial communities to address general questions in ecology and evolutionary biology. But before we move into that, so why should we care about microbes, first of all? So microbes are very important for life on Earth. So they perform ecosystem functions that are very relevant. And one of them, for example, could be the decomposition of organic material. And we know that this ecosystem function cycles carbon to Earth ecosystems. And we also know that many of these very important ecosystem functions for life really emerge from the interaction of multiple species working as a collective. The other thing we know is that now we are facing a lot of stresses in the environment, things like climate change, but also like more uses of antibiotics or pollution. And then one question would be like, how are these functions going to persist in the face of these environmental stressors? But before we can make prediction about function, first we actually need to understand something even more basic, which is how does microbial communities, how do they respond to environmental change? And even though that sounds very simple, it's actually not, because microbial communities are composed by multiple species. So we need to first understand the physiological responses of microbes to environmental stressors. Then we need to understand the population dynamics, so like genetic changes and how they evolve in responses to these changes. And then finally, we need to consider that interaction with other species may be shaping physiological responses, but also population responses. So to deal with a little bit this complexity, what we are doing is to isolate individual members from these natural microbial communities, assembling them in simplified communities, also called synthetic communities, that we can bring to well-controlled conditions of the lab, where you can study them with more control. And our hope is that with those simplified systems, we can get general principles and understanding of microbial communities from nature. So this is the bottom-up approach that we've been using at UC Irvine in my lab. And we are overall interested in understanding the eco-evolutionary dynamics in microbial communities. So there's two big areas of research we're interested. One is, first of all, how do evolutionary responses to stress influence community function? So this is the arrow that is from evolution to ecology. But also we're interested in the reciprocal feedback, which is, for example, how the species interactions can influence evolutionary dynamics. So kind of the eco to evo arrow. And today I would like to talk about two different stories which correspond to two different systems that we're using in the lab. One of them is a three-species community of polymer degraders that are important for ecosystem functions. And the other one is a two-species consortium of cross-feeding bacteria. And they are both these studies in the context of emergence and maintenance of diversity in microbial communities. So the other thing about microbial communities I find very interesting is that they are extremely diverse in terms of just the mere number of species. So many of them can hold thousands of species. And to the point of view of an ecologist, that can be very interesting. So what I'm showing here to illustrate what I think is interesting kind of question is, this is showing a biofilm growing on the shower hose of a domestic shower. So if you cut a domestic shower hose, you can see all this array of like microbes. So these researchers at ETH Zurich, what they did was to take this picture of the biofilm and color all the different species with different colors. So in such a simple environment, which is basically just drinking water and then just like basically a surface, you can see coexistence of all these different types. So the question is, why not one single microbe, the best competitor, overtakes the population? How can we explain that we have such array of species in a very simple environment? And the simplest answer is that, well, actually what we think is an homogeneous environment is not, and there might be different niches where that somehow can promote this coexistence of species. So there's very important studies studying the spatial structure, how that can actually facilitate coexistence, but more than that, it can also promote evolution of diversity. So there's this very famous study of Rainey and Travisano, where they just had a Pseudomonas culture that they basically, the only thing they did was to stop shaking it in the lab and leave it in the bench for a couple of weeks. And then when they came back, they saw from one single ancestor that had one morphotype, they diversified in three different morphotypes. And then they saw that they could colonize different parts of the vial. So just a very nice example of how this can emerge because of the opportunity of having microenvironments. And the same is true with temporal structures. So we know that the environment is constantly fluctuating, and then in one condition, one thing might be more favor, and then another condition, another microbe may be favor. Overall, they balance each other and there's more coexistence. And finally, the one that I'm really interested in, like the role of species interaction. So we know in microbial communities, there's the presence of one species can actually provide ecological opportunity to other species. And cross-feeding interaction are a very clear example of this. So what is, for example, a waste product of one microbe could be actually the food source of another microbes. So there really are actually open opportunities for these microbes. So in terms of knowledge gap, I think like all of these have been well studied, but then there's still a lot of emphasis when we are, for example, thinking not just species interactions on things like negative interactions. So there's a lot about competition for resources, and there's not a lot of information on positive interaction. So how like cooperative behavior could actually influence this, except for the case of, I mentioned, of cross-feeding. So for this talk, I will be mostly talking about like the role of positive species interaction in this coexistence of genotypes and species. So first, the two questions I will be addressing is, how does substrate complexity influence coexistence of leaf litter degraders? And then what role do commensal interactions play in the formation of new bacterial species in communities? So let's dig into the first study. So this one, I started introducing already about like this important ecosystem function, and we will be talking mostly about polymer degradation. And this is really the concept of like when you have leaves from trees and grasses, when they fall into the ground and they are dead, so this leaf litter, we have that they get colonized with a lot of microbes from the environment, and these microbes, what they do is to really break down all these polymers that are in the plant material. And they do that through extracellular enzymes that they release into the environment. In the process of doing all of this, they are actually respiring. So they are like us, and they are respiring carbon dioxide back to the atmosphere. And in just terms of like microbial activities, they are actually big contributors of carbon dioxide to the atmosphere. So now with all the discussions of climate change, a big question is, if these communities are being affected by climate change, are they going to respire more or respire less? And does that mean they are going to amplify or exacerbate the effects of climate change? Or are they going to buffer actually carbon emission and making things better in terms of climate change? So this is the kind of questions that we're interested in. And I joined a collaboration at UC Irvine with Steve Allison, Kathleen Tresider, and Jennifer Martini and Adam Martini to really address this question. And they had a very beautiful system in Southern California, which is the Loma Ridge Global Chain Experiment. So what they've been doing is for more than 12 years, they've been studying these leaf litter microbial communities and then studying how they respond to simulated climate change. So what they have is this, for example, I'm showing here drought structure, where they actually cover and prevent rainfall, and then they can study responses of microbial communities to simulate the drought. And then they have the control plots where they are not doing any manipulation. So the question is, again, how are they going to respond to stressors? And even though they have a lot of information on this, I was a little bit like, I thought an opportunity for me to come into this collaboration is gain more mechanistic insight on how they are actually doing this degradation of polymers. So what we did at UC Irvine with 3 amazing undergrads was to actually, they took over this idea of like building a synthetic community from the leaf litter microbiome, a simplified community. And it was a huge task because really was building a lot of combination of strains and seeing what can grow in the lab. So it really took a big effort. But we did have like some criteria of like what we consider would be important for a synthetic community representative of the leaf litter. Mainly, we wanted to have representation of the major families from the environment. So what I'm showing here is work from Alex Chase, which they have metagenomic data where they have the relative abundance of the different, in this case, phyla from this grass litter. And then, luckily for us, it's relatively simple at the level of the phyla. So there's mostly 3 phyla, which is Protobacteria, Actinobacteria, and Bacteroidetes. So we end up selecting a subset of isolate from each of these phyla, and then we end up getting the ones that are good grower, fast grower, and then that will also coexist in the lab. So this one end up being like Erwinia, Cortobacterium, and Flavobacterium. So just we were also very relieved that this one in particular, Cortobacterium, is actually, in terms of just like a genera, like a species, is a very abundant species in the grass litter. It's up to 10% of all, as a genus, is 10% of the whole diversity. So we think like it's very important kind of like player in this grass litter. So now with this synthetic community, the other criteria that we had is that we can distinguish them visually. So they form different colony size, shape, color, like many different things. And that one was nice because then we don't have to do sequencing all the time. Even though we do have their genomes, we at least can like do a lot of experiments and just tell them apart by just looking at plates. The other thing we had to simplify is how we grow them, the substrate where we grow them. So we can actually think about a piece of leaf litter, in particular grass litter, as just like main polymers, and they are mostly composed of cellulose, hemicellulose, and then lignin in terms of polymers. So we were very interested in understanding, we went to the route of like studying more hemicellulose, and in particular silane, which is the second most abundant polymer in nature. And we also verified from the genomes that they actually had the enzymes to be able to degrade this polymer. So they have this enzyme that can, the silanases that can degrade this backbone of silane. So then what we do is to then grow them in minimal media, now very simplified, and just supplement them with just silane, which is just like a bunch of silos together. And we can also do the experiments with just silos. And the question they were very interested, I was very interested, is a very simple one. We were just wondering what is the influence of substrate complexity for coexistence of species. And for substrate complexity, what we mean is a complex substrate is a polymer because we need extracellular enzyme to break the bonds. So it's not that simple to just uptake it. And a simple substrate is things that you just need to transport it and you can directly take it from the environment. So then the question is, how does coexistence vary depending on these two substrates? And the hypothesis is that polysaccharide might promote species coexistence because things are slower and also they have to release extracellular enzyme, while monosaccharides promote competitive exclusion. And this is just basically resource competition in one case. And the other one, there's many different mechanisms, synergism of enzyme, but also just resource partitioning if there's branch parts of these polymers, or as I mentioned, just slower growth. And it sounds a very simple hypothesis, but surprisingly, it has not been tested a lot in lab settings. So another detail about our experimental setup is that we grow this in batch culture system. So what that means is that in a cycle of growth of these bacteria, at the beginning, they have a lot of resources, they grow exponentially, and we know they completely saturate the resources. This happened for these strains in 2 days, 48 hours. Then we transfer 0.1% of the population into a fresh medium, and then we continue these cycles multiple days. So at the end, what we get is a graph like this that worth explaining because I will show a lot of these graphs. In the x-axis, we have the time in days, and in the y-axis, we have the cell density, which we estimate as colony forming units in agar plates. So then in this particular example, the blue type is able to sustain a serial dilution regime. So we have like this flat line over time. And then the red type, even though it's growing a little bit, is not being able to sustain the serial dilution regime. So it goes extinct. And this is just like just an illustration. So now let's look at the data for these bacteria. Oh, and then importantly for interaction, we can do these experiments growing the bacteria in isolation, so monocultures, or we can put them all together and do these co-cultures or community. And then the idea is to compare the slopes of these things, growing in isolation versus growing with the community, to assess the impact of others in the community. So when we do these experiments, first then growing them in isolation in the xylem, they all can grow in xylem, so they achieve high densities over 10 days. Now, if we look at when we grow them all together, we have that they are also coexisting all together in the xylem. They have the same bacteria, like cell densities in monocultures and in co-cultures. So in that sense, there's neutral interactions, or there's no effect in the community on growth. So now if we look at the monomers, so in the monomer silos, we have that they also all can use silos as expected. And then now when we actually put the 3 of them together, we have now that one of the species suffers from those interactions and it goes extinct by the 10th day of the experiment. So it's one case where we have indeed 3 species can coexist in xylem and only 2 can coexist in the silos. So we have some support of our hypothesis. What we still don't know exactly is what are the mechanisms. So we do have a mathematical model that shows that in the case of silos, resource competition is actually enough to explain the dynamics of one species going extinct. In the case of the xylem, we still don't know what are the actual specific mechanisms. It could be a mix of many things together, but we're exploring a little bit more that. And then I think the most important is that the exciting direction is that we actually found conditions where things coexist. And that's just the beginning of way more that we can do with this system. So now we have even a 4th species in our leaf litter community. So we have Pseudomonas that now we have that they can all coexist, and they can all coexist now in, for example, in leaf litter teams. So what we now are doing is like going back to the complexity of the substrate. So we just do a T of leaf litter, and then we're growing them directly on that. So it's a little bit more realistic, and we have that the 4 species coexist. So now our next step is to actually evolve them under heat stress, simulating, again, one of the stress of our climate change, and see how the function of leaf litter degradation changes, like in the evolved community compared to the ancestral community, and if they are emitting more carbon dioxide or less carbon dioxide. So exciting kind of future direction. I would be happy maybe in 2 years to talk about that. We're just starting these experiments, but it's exciting. The other one that is very exciting is we have a new grant in collaboration with Steve Allison and Kathleen Schuttider to study cross-kingdom interactions. So fungi are actually main kind of players in this leaf litter system that we've been ignoring so far. And so we're really interested in understanding if fungi bacteria together adapt better to heat stress, in particular extreme warming or like heat waves, compared to like just species evolving on their own. So also more work on that will come in 2-3 years. And we are starting these 2 evolution experiments in the lab. So I will be happy to talk more in the future about that. But for now, I would like to move to my second system because it's a very nice system too. And this one is really incorporating now the evolution part, and is how do commensal interaction play in the formation of new bacterial species in communities. So going back to our slide of the mechanisms of basically maintenance of species, but also closely related genotypes, another knowledge gap is that a lot of studies have been done, for example, when you study spatial structure or temporal structure on single species system. So we have like very nice studies on evolution, but like it's mostly studying these monocultures. On the other hand, we have a lot of studies on communities, but they tend to be more like on the short time scale, so on ecological time scales. So I think what's missing is a little bit the integration between all of these and really getting at the eco-evolutionary like feedbacks and how does that really influence appearance and maintenance of species diversity. So the system we're using is, this one is different from the leaf litter. This one is composed by Acinetobacter and Pseudomonas, so only 2 species. And these 2 species are very interesting because they have this unilateral cross-feeding interaction where one feeds on something, excretes a byproduct that the other one feeds on. And a little bit of background on these communities, I started working on them in Switzerland, and they were originally isolated in Denmark. So they actually are 2 species that were coexisting in the environment, and then they were isolated from a polluted soil, which is a creosote-contaminated soil. So what I'm showing here is the image of where these things were isolated. So basically, the soils of this gas work industry. So nowadays, they don't have this kind of like industry anymore. So they don't produce natural gas in that way anymore. But the soils are remaining, and they're quite polluted with a lot of these compounds. And I'm showing a list of these compounds that are in these polluted soils. And hopefully you can see that some of them are actually like carcinogenic or can create health problems. So really a big challenge for many of these cities that have these polluted soils is that we want to decontaminate these soils before they reach the water distribution systems, and that can cause problems to the local population. And in the US, there's also cases of these kind of pollutants that might affect populations. So the idea is to turn into bioremediation and use microbes to actually break down and degrade these into lower compounds that are less toxic. But luckily, in these cases, they didn't even have to put microbes. They were already enriched with microbes. So once they went to the field, they saw there's a lot of microbes that are already degrading these pollutants. And then in particular, there were a lot of microbes enriched for degrading aromatic compounds, for example, benzyl alcohol being one of them. So going back to these 2 species, what we have is that Acinetobacter johnsoni has this ability of breaking down the aromatic ring of benzyl alcohol. And we have that in this system, Pseudomonas putti, that has been engineered to not being able to degrade this aromatic compound. And then that forced then a cross-feed interaction because Acinetobacter degrade the benzyl alcohol into an intermediate product, which is benzoate. And then the benzoate builds up in the cell just because of the cellular machinery is not like exactly all tuned. So there's excess of benzoate inside the cell, and it ends up passively leaking in the environment. And then there, Pseudomonas can actually cross-feed on these benzoates. So we have this cross-feeding interaction. So what I did in Switzerland is just to actually trying to see a visual of how these bacteria are in a microfluidic chamber. So what I'm going to play is like just a time-lapse movie where I have Acinetobacter as these round gray cells and Pseudomonas as the green fluorescent, more elongated. So you see at the beginning, there is mostly a growth of first Acinetobacter, and then this is followed by this like green fluorescent. So there's this sequential growth, which will indicate that first Acinetobacter has to grow, and then this is followed by Pseudomonas. Then in terms of like quantifying things of ecological interaction, you're already familiar with the system. So these grow faster, which is nice. They grow in 1 day. And then we dilute them 1% over these passages. And we know for sure from other experiments that they exhaust all the resources after 24 hours. So then how does it look with the data? So when we do these kind of experiments in isolation, we have that Acinetobacter, as I mentioned, is very good at using benzyl alcohol, achieve high density. And then Pseudomonas is actually persisting in the population, but at low density. So we know that actually this persistence is because they are able to grow on residual carbon sources that are very hard to minimize, but they are really in low density. But the important thing to see is that now when we grow them together in co-culture, Pseudomonas achieve 2 orders of magnitude higher growth in the presence of Acinetobacter. So again, we are seeing this like beneficial effect of having Acinetobacter. So in terms of ecological interaction, this is really a commensal interaction where Pseudomonas is highly benefited by the presence of Acinetobacter, and Acinetobacter is actually not really affected by Pseudomonas. Maybe just a little bit because it's like actually cannot use the like extra benzoate, but overall we have a commensal interaction in this system. So now what we wonder is how stable are these kind of systems over evolutionary timescale? So we know they're pretty stable over 6 days. So that's, we consider is still ecological timescale. So what would happen if we actually evolve this system for longer? Are they going to persist? And is this interaction going to break down? What happens? So to address this question, we did an evolution experiment where we actually propagate this one for not 6 days, but a full month. And this is equivalent to 200 generations for these bacteria. And we did these experiments for monocultures and then the co-cultures with 4 different replicates. So trying to assess how replicable is evolution. So what do we see? So we see that in benzyl alcohol of evolutionary timescale, we have very similar dynamics from the ecological dynamics. The Acinetobacter persists over time, and then Pseudomonas too, but at lower density. So now see how the x-axis is over 30 days. And I'm plotting just the result of one replicate because that's the one that we follow. We don't have data for the other replicates, but I'm plotting here the endpoints of the other replicates. So nothing too exciting about monocultures. So when we look at the co-cultures, we see that there's also coexistence for the Foley experiment. There's interesting more fluctuation in dynamics. We don't know exactly what's causing this. There's this huge like major disturbance that happened in Acinetobacter, which also affected Pseudomonas. But like overall, they recover. Again, we are not sure what happened there. We're not sure if it's something intrinsic of the system or it was an external perturbation. Sadly, we were not tracking the other replicates, so we don't know. It would have been interesting to like actually track the other ones to see if these kind of things are common. But at the end, for the 4 replicates, we have coexistence. They both managed to reach until the end of the experiment. And there's slightly higher densities of Pseudomonas with Acinetobacter than in monoculture, even though it's not noticeable. But overall, we can say like, okay, these communities are, interaction seems to be quite stable over evolutionary timescale, just in terms of like nothing outcompeted anything. So that was good, but like still not maybe super exciting, except for something exciting. So what we did observe is that Pseudomonas actually diversify into the 2 different morphotypes. So when we were doing these experiments, very quickly we noticed that there was this appearance of this larger phenotype compared to the ancestral phenotype. So we have like large colonies and small colonies. And I think what was very exciting is that this diversification or dimorphic population only occur when we evolve them with Acinetobacter. So it was only observing co-cultures. In monocultures, we pretty much have like only large colonies. So it's very interesting kind of behavior. And then here I'm showing, since we have like the data resolution for one of the replicates, so I'm showing here now like a zoom in detail into what happened to Pseudomonas. So the ancestral morphotype is in dark blue. And then I'm plotting here actually the dynamics of the large morphotype. So we can see they appear quite quickly, like after 4 each generation, and they start increasing in frequency. And in monocultures, they actually fix in the population and they outcompete. So this is like the ancestral morphotype goes extinct for the monoculture. So now if we look at the, and then that's why we end up with only large colonies. So now if we end up looking then the one with Acinetobacter, we see also appearance of these large morphotypes quite early in the experiment. The increasing frequency, the difference is they are not overtaking the ancestral population. There is stable coexistence for at least 100 generations. So you can see how they like just persist both of them when Acinetobacter is present, not when Acinetobacter is not there. So that's why we end up with these like dimorphic populations. But we wanted to have a little bit more of quantification about these large and small colonies. So what we did was to try to characterize the traits in these populations. So what we did was to isolate 30 colonies from these populations, and then doing grow curve experiment with a plate reader, where we just follow the optical density over time over a 24-hour cycle. And we fitted many parameters, but the one that is more relevant is the yield, which is basically the final optical density divided by the amount of resources. And from this, we fitted this parameter to 30 individuals or colonies from each population to try to see how the trait evolution changes over time. So this is the result from this experiment. So now I'm plotting the distribution. Now I put these 30 individuals that represent my population. How does the yield look in the evolved population versus the ancestral population? So what we can see is that in the monoculture, there's a shift into having things with higher yield. So it makes sense, is where everything was big colonies in the plates. And it's actually huge because it's three-fold higher yield compared to the ancestor. And the whole population is shifted towards higher yield. Now, if we look at the co-cultures with this quantification, we have a very nice, significant bimodal distribution where we have a subpopulation with this threefold increase in yield. But we do have a subpopulation which has a yield pretty much the same as the ancestor. And that was very interesting. But of course, a grad student, very talented grad student in my lab was wondering, is this based on genetic basis, or is it just phenotypic things? So what Zara did was to actually sequence this, so taking a small morphotype or a large morphotype, and also whole populations to see if we can see mutations associated with these morphotypes. And just a little details of how we call mutations. So basically what we know that we start with a clonal population. So they don't have recombination, they don't have plasmid. So basically, we align these genomes of the ancestor and the evolved clone, for example, to a reference genome. And any variation or any change that occur only in the evolved clone and not in the ancestor, that's what we call de novo mutation. Again, we didn't start with standing genetic variation. We assume it's all clonal from the beginning. So these are the kind of mutations we're looking at. And then what we observed was very nice because it was a very straightforward case. So we did observe that in the ones that have higher yield, there was one mutation where fully fixed in the population at 100%, which is a prem-shift mutation in the FLQ gene. So the FLQ gene is a master regulator of flagella and motility and biofilm and many traits. It's highly pleiotropic. And then we have these mutations. Not only this particular, we see a lot of replicable evolution or convergent evolution. So multiple lines have this mutation, and they are all inactivating this FLQ gene. So that was very neat. And then we see that in the co-cultures, we also see these FLQ mutations. So again, like a high level of convergence or parallelism, but they are not fixed in the population. So we find them at 36%, which again, we know now with isolating single clones and sequencing that the FLQ mutation is really causing the higher yield in these strains. So why would this be advantageous? So we know actually mutation in flagellum is actually quite common in evolution experiments. They occur in many labs, not only in our lab. And then the idea is that these kind of mutations save plenty of energy in a well-mixed environment, and then they can redirect that energy for higher growth and yield. So to get a little bit more of support for these possible hypotheses, what Zara did was to do swimming assays. So now what I'm showing here is some assays in soft agar. So basically here what we have is that the ancestor, we spot it in the middle and then we let it swim in those plates. So this halo around this initial spot is actually how much they swim over 48 hours. So we know that the ancestor of Pseudomonas is actually motile and they can swim. And then when we do this with these FLQ mutations, we actually have that they cannot swim, they don't form this halo. So then basically we confirm that they cannot swim, but also we did biofilm assay, and then we also know Pseudomonas is very good at forming biofilm, thick biofilm. And then when we do biofilm assay with these FLEQ mutants, they also have a significant reduction in biofilm formation. So it seems like there's definitely a lot of traits that are being affected by this FLEQ mutation. So it all makes sense. That hypothesis that they are saving energy because they can, they don't form biofilm, they are not swimming, makes a lot of sense. The question is then what's up with this subpopulation and do they have mutations? So they do have mutations. We know they are not identical to the ancestor, but those mutations tended to hit genes that are not well annotated and we don't know exactly the function. And they are also low frequency mutations. So there's plenty of zero mutations, but like they are around like 10% in the population or even less. So we don't know exactly what they are doing. So the big question is, why would those more morphotypes, which are not identical of the ancestor, persist in the population only when Acinetobacter is present? So here's our hypothesis. So we put forward the foraging yield trade-off hypothesis. So basically, there's the part of the FLEQ mutation that are coexisting with this other one. So our hypothesis, as I mentioned, is the FLEQ mutant have an advantage because they save energy, they can have high yield. So that makes sense, they persist in the population. In terms of the other ones, what we think is that given that they retain the ability of swimming, there has been a new research that shows that actually motility can actually facilitate partner encounter just by like bumping, like by being motile, they can actually bump into the partner more easily. It's not chemotaxis, it's just like more probability of encounter. And once they encounter the partner, they actually can attach and adhere better to the cross, like in this case, to the species that cross this benzoate. So then we think that something like this might be happening, and then they are extracting a high energy resource, which is the benzoate. So then there will be advantage of retaining this ability of swimming only when Acinetobacter is present. Otherwise, they will be outcompeted by the FLEQ mutants. So to get a little bit of support for this hypothesis, then we have the swimming assays for the small morphotypes or clone, and we see that they retain the ability of swimming. So we know they are still motile. And also, they actually also have biofilm formation. This is not significant, but now we have new data showing that they actually form more biofilm compared to the ancestor. So definitely there is some support for this hypothesis, but we need to do more experiments. We're doing microscopy and more things to get support of that. The other thing we were not satisfied really with our system is, well, how important is that the benzoate is excreted by Acinetobacter versus externally supplied to our media? So what we did not do in our original experiment, because we didn't know this was going to occur, is we did not have a control where we actually supply benzoate into the media. So it was just benzyl alcohol. So we don't know if it's just because having extra benzoate that actually we have this diversification. So to try to understand how important it is that this benzoate is produced by Acinetobacter, we did a new evolution experiment funded by NSF. And this one was a large-scale evolution experiment. So luckily, a postdoc, Thibault Rosasa, came on board because these really are laborious experiments. And the new evolution experiment now is like fully factorial, very well designed. So basically what we have now is that we have a condition where we have species in isolation, so monocultures of Pseudomonas where we add external benzoate. And also we have the condition where the benzoate is produced by Acinetobacter. And then we also have a condition where we actually have Pseudomonas with benzoate produced both by Acinetobacter, but also supply into the medium. And just to have the full factorial design, we have the Pseudomonas with higher amount of external benzoate. So in this case, we can kind of consider that the benzoate could be like in two different sources, which is two types of kind of cross-feeding. One is the obligate cross-feeding, where Pseudomonas has to rely on Acinetobacter to get this benzoate, and another one is more like a facultative cross-feeding, where they can get it from both sources. Then we increase a little bit more like the scale of this experiment by including 2 more replicates, so 6 replicates now. And we did 200 generations. Now we have 1,000 generations, but I will only show the result of 200 generations. So you are familiar now with this distribution. So let's look first at the monocultures. So similar to our previous evolution experiments, we have basically in both of the conditions, we have a significant shift into the yield, the distribution, into having higher distribution in the monoculture. So very similar to our other evolution experiment. In the facultative cross-feeding, what we have here is that we also observe a shift in the distribution, but it's not as significant as in the monocultures. But there's definitely a little bit of a shift of the distribution. Now, in the condition of the co-culture, obligate cross-feeding, first of all, we were very surprised not to see like massive amount of diversification, like our previous experiment. We only have one case of significant diversification, but in this case, actually, most of them, they don't have like these two subpopulations. But I think what you can notice is like, what's very interesting and different from this obligate cross-feeding is that they have a distribution that is relatively similar to the ancestral, if not shifted towards the other direction of having lower yield. Only in the obligate cross-feeding interactions. So this again is very preliminary results, but we also sequence whole populations of these 200 generations, and it's a complex figure. So I'm just first going to present one panel. So what we have here now is the mutations that are in the population. So these are the most frequent mutations. So there's plenty of things that are not well annotated. And then what we have here in the x-axis is the 6 replicates that we have. And then we have the mutation frequency with shades of blue. So the darker, the blue, that means it's like a fixed mutation, 100% in the population. And then we have like the low, like the light blues are like mutations that are high frequency, but like still like 20%, so lower frequency. So the first thing you can see is that there are parallel mutations, things that occur like independently in different lines. So those are the mutations that for us is interesting because it would suggest there are adaptive mutations. So now that we have seen this one, then I want to show all the conditions. So these are then for different conditions that I mentioned. So the monocultures here in the top, and then the co-cultures, the facultative cross-feeding and the obligate cross-feeding. So there's a lot going on, but basically like one thing we can look at is go back to our FLEQ mutation, which was the one that was interesting. And then we were reassured and happy to see them reappearing again in the monoculture at very high frequency. It's fixed for many of these populations. So this is very similar to our previous evolution experiment. But I think what's even more exciting is that if we look at FLEQ mutation for the obligate cross-feeding and facultative cross-feeding, we pretty much don't have mutations in this FLEQ mutation. But overall, if we look at actually all the mutations that are related to flagellum assembly, motility, and regulation as a pathway, there's a significant underrepresentation of these mutations. So there's absolutely no mutations for the ones that are obligate cross-feeding. There's very few mutations for these other, where Acinetobacter is present, and there's mutations in the monocultures. So basically what this is telling us is that all these functions related to like motility, biofilm formation, all these things seems to be relevant only when Acinetobacter is present. So putting these together, the conclusion is that across the species interaction, we saw a case where you can actually maintain coexistence of two different types. So that was the first evolution experiment. So big influence of having another species for different evolutionary outcome. And the other thing with the new experiment is, I think what's exciting is that we should rethink what we think about essentiality of genes depending on their environmental context. So in this case, flagellar gene seems to be very essential only when the cross-feeder is present, even in a well-mixed environment, which can be surprising because one would think that in those well-mixed environments, structure would not matter. But in this case, it does seem to matter a little bit. So just putting all together, what I want you to get out of this talk is, of course, the context matter. You know this, you are EEB department, so this is a no-brainer. But I hope I show you like some evidence that we know now that species interaction, they can play an important role shaping evolutionary dynamics. So we definitely should study evolution in a community context. And also I show a case where like maybe we should also be thinking about like, for example, gene essentiality in a community context, because interaction can have a lot to do with what we consider essential and not essential. And with that, I would like to thank, first of all, like my amazing lab, which has been contributing quite a lot for this research. So particularly Parmis and Zara, who led the two projects I mentioned, and Thibault now, which is on board and helping us with all these experiments. All the people in Denmark and Switzerland, which helped the first part of the evolution experiments. And then a lot of really amazing colleagues at UC Irvine that has been very supportive. And I'm just underlining the formal collaborations and all the work that's presenting. Many of these things are in collaboration. Finally, the funding agencies, especially like the two new NSF grants, which is quite exciting, and UCI Bio. And you, of course, for your attention and for being here today to listen to me. Thank you so much.
First off, really cool talk. I really enjoyed hearing the two experiments. And I was wondering so one striking thing about particularly the kind of second set of experiments was that in the monoculture, the Pseudomonas, the mutation allowing for the large colonies fixed, whereas in the co-cultures, it was at intermediate frequency. So I'm wondering, like, from that, I'm taking, like, in this particular community context, it seemed like the monoculture was supporting kind of a directional selection, whereas it's balancing in this kind of community context. I'm wondering if you have a sense about whether like the complexity of community, does that tend to shift evolution towards more of like a kind of balancing selection regime, or do you have like a sense of whether that's kind of a generalizable finding?
Yeah, no, definitely. I mean, I think there's a lot of talk of like diversity promoting diversity. So this idea that actually like you will have more balancing selection in communities because of the community context. So I think that is an overall thinking in the field right now. In our case, definitely like, I mean, we have a little bit more of mechanism, but yeah, the idea is that the other species are doing something. They are doing, I think, just more than the cross-feeding. It seems like there's also just they are modifying the environment. We don't know the eco-evolutionary dynamics, so we're moving into actually seeing what happened to also Acinetobacter, because we have been ignoring it so far. And then we know that like the way they change might actually change the environment, we might change the species. So there might be very interesting dynamics, with more potential of being this kind of balancing selection, where like we have more maintenance of different ecotypes or like genotypes in the population. But yeah, it's a great question. I think it's an overall thinking in the field.
I know they found that with like fluctuating temporal conditions.
Right.
But,
I mean,
so it's interesting that there's not that.
yes.
I don't know.
Yes.
I have more questions, but I'll let other people.
Again, great talk. It was also amazing to see. I saw Zora's talk in January at meetups. It was
Yes.
So
Exciting.
I think the species interaction, right, that's one thing that's clearly modulating the evolution. But another thing, right, is firstly in the serial evolution experiment, like when you say it's well mixed, is it like shaking?
Yeah,
right?
definitely important question.
So you're shaking the
Yeah, I forgot to mention, and I think this is very relevant. So the shaking is 150 rotation per minute. So it's not the super shaking of 250. It's not like the low shaking, considered low shaking. So it's kind of intermediate, which seems to be very critical.
Yeah, so that's exactly the point, because the soil is obviously as little, these are derived from soil, right? So that's as little shaking as possible, right?
Yes, exactly. Absolutely.
So I was wondering, like, if you had considered possibly like altering the shaking frequency to see if the evolutionary dynamics change.
Yes, absolutely. So Zara's third chapter is going to be about that. Exactly. So we are going to do zero shaking, 50 rotation per minute, 150, and then all the way to 300, I think. because we want to know exactly like how important, I think we were lucky, like to find like the right, like just out of luck, the right kind of like ear shaking that would like promote still some structure and interesting happening. We believe that actually if you go to the too extreme, you might lose the effect. So if you go strictly static, there might still be coexistence of things. There might be biofilm, there might be very interesting things, but maybe different kind of mutations. That's what we observe. And the other extreme of super shaking, maybe everything will like break down. So
Yeah, but you know, initially, right, it's coming from the soil. Right?
Oh,
so initially
yeah.
there's no shaking at all.
And initially
there,
there's, yeah,
flagella
yeah.
is always present, right?
Yes, that's right. Yeah.
Okay, well, I have one more.
Yes, of course.
So in a flagella in the soil, do you have any idea of like how far you can diffuse with a flagella if you're in soil?
Oh my gosh, that's a great question. I don't know. To be honest, we don't think too much about soil. Well, we should, but like I really don't have a good feeling about like the complexities of the soil and like the micro scale of things. So I would not be able to really
It's just
keep
the
inside.
diffusion and adhesion
Yeah.
is such an interesting potential.
It's super interesting. Yeah, the other thing we're also doing is like going back to just structure, not maybe soil particles, but just biofilm. So the postdoc Thibault is doing just biofilm essay where we just have them like in a glass slide and see how they are actually having different interesting behavior. And it seems actually that the ones that are still motile, they do more than just move. They seem to be hypermotile. So this is like a very preliminary observation, but they move way more than the other ones. They are like completely crazy in these kind of experiments of more like glass slide. So there's definitely something going on there. We don't know exactly what. More to come, I think. Great questions. Thank you. Yeah.
I have just a couple of questions. I'm interested in your like fungi hypothesis.
Oh, yes.
Because fungi also have like a lot of like enzymes, right?
Right. Yes.
So is the idea there that like, you know, this is microscopic or more microscopic organism and initially degrading some of these like complex polysaccharides and opening new niches, like you're hoping to find like this more, it actually is opening up niches or like actual positive interactions with that?
I mean, that's a great kind of hypothesis. Ours was not that one, but there's definitely very interesting things that can happen, synergism with the extracellular enzyme.
Yeah.
Or particular hypothesis for the NSF brand was biofilm formation. Yeah. So the idea that some species might form biofilm, others don't have the ability, but then by being all together, they can take advantage of the gooey biofilm that would protect from drought and heat and things like that. So that's our mechanistic hypothesis of why they will do better in a community than on their own.
And then your takeaway from that last experiment about like genessentiality and like environmental context is reminds me a lot of like TNC experiments as well, where you knock out like individual genes.
Oh yes, I know. Yes.
Yeah. So I'm wondering, like, have you thought about like using your two environments, the cross-feeding and the like monocultures in a TNC and actually like quantifying the fitness effects of like every gene?
Yeah, so that's a great thing. So like just the word, like the technique itself. But yes, so I have like a incoming postdoc, which is like super motivated to do that. He's a bioinformatician. He has never done experiments. So I'm like, how on earth are you planning to do that? But like, we definitely want to do it because that would be amazing to do like through like just knocking out, like seeing like what would be. And I think, definitely that would be super exciting. So hopefully we can do it. I'm not sure yet. But like, I will let you know if we do it. Yes, of course.
Just had a question. You guys looked at like the viscosity is playing a big part of this as well. Definitely more realistic kind of systems if you're seeing changes in viscosity, potentially can bring or maybe facilitating kind of that loss of the flagella and it's low viscosity, it's able to move around without much energy loss regardless, it doesn't matter.
Yeah.
Versus like a high viscosity system.
That would be great. We have not thought about it, but I think that sounds something easy to implement, but that would be amazing because definitely like that's another way of playing with structure. And I think it might have like a big element on all these dynamics for sure. Yes. yeah, it's great. It's great. Yeah, definitely thing to think about. Yes, of course.
So you mentioned early on about like you mentioned Peter Chess and coexistence.
Oh,
theory,
yes,
the model.
yes.
So given the results you had here, do you think that's predictable from sort of traditional coexistence theory in ecology, or do you think you would have to add in more about like commensal interactions and interactions with stressors to really understand what's going on here?
Yeah, that's a great question. So once you go into the details of our system, like it's a little bit annoying because we did like all this like very coexistence theory of invasion experiments from clones and trying to see test the theory. And we actually didn't get very far with single clones. So there is something about like, it is maybe predictive at the population level. Once we try to do like very like simplistic experiments with clones and this, like the invasion experiments in the presence and absence of Acinetobacter and things like this does not work. Like
Doesn't match the theory.
it does not match the theory. Like the large morphotype always overtakes. So then like we are like, okay, that didn't work. So we think like it's really like then we need to take the whole genetic diversity to actually have these things like matching the predictions.
So I guess it's like one of the.
Yeah, definitely. Such a nightmare, yes.
Yeah, I was wondering for the evolutionary changes that you caught in the small colony,
Mhm.
so the evolved small colony population,
Mhm.
do you think that those are adaptive or just like neutral changes? And then I also noticed that there was like later in the presentation you had the 4 different regimes like co-colonized. There was like a strip of things that were actually fixed across all of them, and I'm wondering if there was any interesting story there.
Yeah.
Well, I guess not. Yeah, I guess it's unannotated. So.
Yeah, I mean, definitely. I think there is interesting adaptation overall of the media condition, like that are independent of Acinetobacter. Here, though, like what I was talking about, like standing genetic variation, we have one mutation that was. That's why it's in everything. And this one went extinct. So it's not there because we had one case of extinction. Yeah. So there's definitely a lot of other interesting things to look at. And there's also mutations that are particularly just in the cockroaches and they are not here. So I think like we just got this like a month ago. There's still so much we need to like do to try to interpret what's going on. But it's great because like this kind of experiments always deliver so much for years to like analyze things. So already the big effort is done in a sense, and we're happy about it. Now it's like all the fun of like trying to interpret this data and making sense of things.
Like, do you know what the UVRY, the very bottom is?
Yeah, so it's like a sensor response protein. So like as far as like the annotation goes, trying to understand, like we still don't know. I mean, we have not formed an hypothesis of what, but definitely like that's one that we are very interested in following up. Yeah, good question. All right, thank you so much. Thank you. Thank you.